So these are just the images from your textbook, but I guess I'll narrate them real quick. So here's the chromatin. So chromatin is DNA with the associated packaging proteins, also all the proteins involved with transcription, right? So remember, transcription is the first step to building the proteins who are encoded in the DNA, that are encoded in the DNA. So DNA codes for proteins. That's what genes are, is protein codes. So there's a lot of proteins involved in transcribing the proteins that are needed constantly by the cell. So, that again, that's transport proteins, enzymes, communication molecules, structural proteins, all kinds of proteins are needed by the cell all the time. So they all have to be transcribed and translated. Transcription happens in the nucleus. So chromatin is the DNA, but also associated packaging proteins called histones, and RNA polymerase and all the other proteins you learned about that are involved with transcription. All right, so then you have, so that's interphase, just a mess of DNA it's not organized, it's all open and accessible for transcription, and then of course translation of proteins. So all the genes are accessible for being read. Then prophase, you have the DNA starts to condense into chromosomes, the spindle fibers begin to form, the nucleus begins to dissolve. The nucleus is completely disassembled by prometaphase. I didn't used to learn this with prometaphase. Prometaphase is just an intermediary step, I guess, between prophase and metaphase. But the nucleus is completely disassembled, the chromosomes are completely formed, right? And then the spindle fibers attach to the centromeres for each of the chromosomes. Metaphase, the spindle fibers, again, those are the marionette strings that orchestrate this whole process. They line up the chromosomes on the equator, and these are duplicated chromosomes, of course, made out of two identical sister chromatids because each chromosome was copied via DNA synthesis in S phase of interphase, right? So these are all duplicated chromosomes. That means each chromosome is composed of two identical sister chromatids. They line up along the equator of the cell, and then they get pulled apart during anaphase. So whether you're talking about mitosis or meiosis, prophase is always about getting things ready. So let's go back to prophase. So whether you're talking about mitosis, meiosis 1, or meiosis 2, prophase is always about chromosomes condensing and spindle fibers forming and attaching. Metaphase is always about lining up on the equator. Anaphase is always about things getting split apart. In mitosis, it's the sister chromatids getting pulled apart. In meiosis 1, it's about homologous pairs getting pulled apart, but whatever it is, anaphase is about pulling things apart to opposite poles of the cell. Again, this is orchestrated by the spindle fibers. Telophase or telophase is often overlaps cytokinesis. So in telophase, the nucleus reforms. That's not shown in this picture from your textbook, but the nucleus reforms, spindle fibers disassemble, and if you were to draw this out, so you should, if you were to draw this out, you would see, if you were to allow the chromosomes to recondense, so the chromosomes decondense at this point because they're going to go back into interphase, into chromatin. But if you were to keep the chromosomes condensed, you would see that each daughter cell now has identical sets of genetic information. So each of these identical sister chromatids got pulled apart in anaphase, so the cell is basically getting divided down the middle right here between those two sets of genetic information, those two identical sets of genetic information. So this part of the cell becomes one daughter cell, this becomes the other daughter cell, and they're genetically identical. So in telophase, nuclei forms around the two sets of genetic information, the chromosomes decondense, spindle fibers disappear. Cytokinesis is about the rest of the cell dividing around the two new nuclei. All right, so that's mitosis.